Translation occurs in three steps: initiation, elongation, and termination. Translation begins with the association of an mRNA, a small ribosomal subunit, and an amino acid charged tRNA, coming together to form an initiation complex. Note that the mRNA start codon is AUG, and the first amino acid of this polypeptide and all other newly made peptides is methionine. The large subunit of the ribosome joins the complex. The ribosome contains three sites for tRNAs. The A, or amino acid site, accepts new tRNAs bearing amino acids. The P, or polypeptide site, is where a tRNA adds its amino acids to the growing polypeptide chain. The E, or exit site, is where the tRNA resides before exiting the ribosome. The ribosome now enters the elongation phase of translation. A tRNA with an anticodon complementary to the next codon of the mRNA enters the A site of the large subunit. The large subunit catalyzes two reactions. It breaks the bond between the tRNA in the P site and its amino acid, and it catalyzes the formation of a peptide bond between the amino acids from the A and P sites. After peptide linkage, the empty P site tRNA shifts to the E site on the ribosome as the ribosome translocates the length of one codon, releasing the empty tRNA. The second tRNA, now bearing a dipeptide, is in the P site. The next charged tRNA enters the open A site, and the elongation cycle repeats. Translation is terminated when a stop codon, in this case UAA, enters the A site. Stop codons do not encode amino acids or bind tRNA molecules. Rather, they bind a protein release factor, which allows hydrolysis of the bond between the polypeptide chain and the tRNA in the P site. The various components of translation then dissociate from each other.